everyone, and welcome to our second week on proteins. Today we're going to discuss first protein biosynthesis, and then we're going to talk about protein biosynthesis and degradation according to physiologic state. So first we're going to start out with protein biosynthesis. And our objectives for this part of the lecture are to define and discuss the components of nucleic acids, discuss transcription and translation in protein biosynthesis, discuss the three different types of RNA, explain the concept of epigenetics and how it works, and list some chronic diseases that could be impacted by nutrigenomics. Okay, so first we're going to talk about nucleic acids. So our nucleic acids are part of our DNA and RNA and are formed from repeating units called nucleotides, or sometimes we call them bases. And a nucleotide is made up of a nucleoside, which has a nitrogenous core here and a pentose sugar here, and then also a phosphate group. And the difference between a purine or a pyridamine nucleic acid is that pyridamines have a six-membered nitrogen containing ring, while the purine has a five-membered and a six-membered nitrogen containing ring stuck together. And here's a table of our nitrogenous bases. Hopefully this looks pretty familiar to you. So we know that specific bases pair with specific other bases. So in DNA, our adenine binds to our thymine, whereas in RNA, our adenine binds to uracil. And in both DNA and RNA, the guanine and cytosine bind together. Okay, so now let's talk about how exactly proteins are synthesized. And to do that, we're going to talk about two processes, transcription and translation. And again, hopefully some of this is reviewed for you. Now, transcription is the process by which we transcribe DNA to RNA. So this occurs with the enzyme RNA polymerase. You can kind of see a picture here, and I have a better picture coming up here. But so this big enzyme right here is the RNA polymerase. And as it goes along a DNA strand, it unwinds the DNA strand and reads one strand of the DNA to produce a complementary RNA. So you can see here, maybe. See here, we're unwinding, and then this is going to be our coding strand, and this is the mRNA that is being produced off of this DNA sequence. So you can see that the mRNA is base pairing with the coding DNA strand. So mRNA, so let's briefly talk about the different types of RNA. So first we have mRNA, which is the genetic coding template used by translational machinery to determine what amino acids are going to be incorporated into a protein. So that this is the one that's made right off of the DNA strand. We also have tRNA, which form covalent attachments to individual amino acids and recognize the sequence on RNAs for insertion of the correct amino acid. And we'll go into more detail of that moving forward. And then we also have ribosomal RNA, or rRNA. And these are assembled with ribosomal proteins to form ribosomes that engage the mRNA and form a catalytic domain so that tRNAs can enter with the amino acids. And again, I have more visuals coming up, so hopefully that'll make more sense. Okay, so here is a picture of what we just talked about. Here's our RNA polymerase. Here's our DNA strand. The DNA strand is being unwound. With these base pairs on this DNA strand, we have complementary base pairing in order to make an mRNA, and then we're going to wind the DNA strand back up with this polymerase. So after we complete this mRNA strand, then the mRNA is going to leave the nucleus, and the DNA is going to remain inside the nucleus. Now, while the sequence of our genes can't be changed, the availability of genes for transcription can be impacted. according to gene rearrangement, amplification, or deletion. Okay, now let's, so in order for genes to be activated by transcription, first, so hopefully this is going back in your mind to biology. Um, in order for genes to be activated for transcription, chromatin modeling must first occur. So remember that DNA is not really organized prior to chromatin remodeling, and then it kind of goes into these like kind of X-looking um, DNA strands or chromatin. Okay, so the availability of these genes for transcription depends on a couple of things. So, first is the acetylation of histones. And remember that histones are proteins that are associated with DNA in the nucleus and help condense it into the chromatin so that we can transcribe it onto mRNA. And um, if we acetylate these histones, then that impacts if the genes are going to be expressed. So here we have, you can see a little graphic down here, and lysine on histones removes a positive charge and therefore reduces the affinity between the histones and the DNA. So if we acetylate the histone, then we are sending a signal to transcribe that gene. Whereas if it's not acetylated, then it's going to not be transcribed. Transcription also depends on the methylation of bases. So if we add a methyl group onto DNA in a very specific location, it will impact if that gene is transcribed. So here we can see that we have our promoter region and a gene that can be expressed, whereas if the gene is methylated, then it stops transcription. So through these couple of mechanisms, we can actually alter if our genes are going to be transcribed or expressed. Okay, now let's move on to translation, which is basically the translation of an RNA into a protein, or more specifically, the process by which an mRNA specifies the amino acid sequence to create a protein. Now, this occurs when mRNA leaves the nucleus and attaches itself onto a ribosome. So you can see here the mRNA is leaving the nucleus and it's attaching onto a ribosome here. Then a tRNA collects amino acids from the cellular fluid. So here's free amino acids. Here's our tRNA. The tRNA is collecting these free amino acids, and they're going to then bring them back to the ribosome and kind of and start to attach in the sequence that is indicated on the mRNA. And in doing so, then the amino acids begin to build on each other and we get a protein chain. Now, you see that in this tRNA, we just have one amino acid, but we have three base pairs. And it's these three base pairs called codons that determine which amino acid is going to be collected by the tRNA and added onto the growing protein strand. So really, mRNA here is ensuring that the amino acids are lined up in the correct sequence. So if we have with the number of nucleotides that we have in all of the different combinations that we could have, we have 64 possible combinations of nucleotides. I'm sorry, this should say 3 nucleotides, not 4 nucleotides. And we have 20 amino acids. So you can see there's some duplication. We have multiple codons that can code for a specific amino acid. However, there's each 3-letter code only is associated with one specific amino acid. It wouldn't be associated with 2 amino acids, for instance. So you can have multiple codons for each amino acid, but only one amino acid for each codon. And here I have a little video. about the advertisement.
Hey, it's Professor Dave. Let's talk about DNA transcription and translation.
He knows a lot about the science stuff. Professor Dave explains.
Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish or a cat or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene. And after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid. But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first two, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. So in this two-step process, DNA is transcribed into an mRNA, and then this mRNA is translated into a protein, all simply by obeying the base pairing that occurs in nucleic acids. And since every gene codes for a specific protein, and proteins make up most of what you are, from your muscle tissue and organ tissue to all of your receptors and enzymes, this is how DNA carries the code for a living organism. Thanks for watching, guys. Subscribe to my channel for more tutorials, and as always,
Okay. So gene segments that get both transcribed and translated are what we call exons. So as amino acids are lining up in the correct sequence on the ribosome, and they're attaching to the mRNA, the ribosome moves along the mRNA and binds the amino acids one after another onto the growing protein strand. And once all of the proteins all of the amino acids have been attached, then the protein is released. tRNAs can be used for building more proteins, and the mRNA is degraded. And I have a really neat little video here to demonstrate translation as well.
What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information off a hard disk on a computer and put it into memory, making it a real program that's running. So that process of DNA to RNA is like running a program, double-clicking on an icon.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
Okay, so that's kind of neat because it shows you the actual speed by which these processes are happening. And you can see that from that video that it's 40 to 100 amino acids can be added to a growing protein strand in just a second. So it's a very, very fast process. Okay, and here's just kind of an overview figure demonstrating what we just talked about. Okay, so again, hopefully part of this is review, but the end of the process that we just talked about is an amino acid chain, and that's called a primary structure. However, proteins can then be folded into secondary structures, and this occurs because of interactions between the side chains on the polypeptide achieved through weak bonds, such as hydrogen bonding. So that's what causes kind of this helix formation. Then many proteins, such as transport proteins, enzymes, and immunoproteins, exist as an even more complicated tertiary structure. And this structure occurs because there are interactions among amino acid residues or side chains that are located at considerable distances on the chain. So then you have the clustering of hydrophobic amino acids, electrostatic attractions between oppositely charged amino acid residues, covalent bonding, and so on. And that's why you get these kind of more complicated folding structure. And then when 2 or more polypeptide chains have bonding between them, such as hydrogen bonding or electrostatic attractions, then they also have interactions, and we get the combination of more than one protein strand, and that's a quaternary structure, such as hemoglobin. Okay, now I want to take a few minutes and talk about epigenetics. I'm not sure if you've had exposure to this concept in your other classes, but this is really getting to be an important factor to consider when we're considering the etiology of disease and how we treat and counsel our patients. So epigenetics involves genetic control by factors other than a DNA sequence itself. Epigenetic changes can switch genes on or off and determine which proteins are transcribed. So what this concept is saying is that, yes, we are born with a specific DNA sequence, but according to exposures that we may have, those genes, the transcription and expression of those genes can be turned on and off. And it does this through some of the processes that we just talked about. So genes can be turned on or off through histone modifications, DNA methylation, and RNA-associated silencing. So basically what that's saying is that the RNA sequence could trigger DNA methylation or histone modification. I have one more video. I know that was 3 kind of right in a row, but I think this is a nice video for explaining epigenetics. Our friend Hank here.
You're finally meeting his family of
Again,
8
sorry for
over a
that.
home-cooked meal big enough for 30. When you So say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, and you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, Clone probably has a tattoo on its face. And if Clone had stayed in the circus, Clone probably, professionally rides a unicycle over a tightrope, so Clone is probably in pretty good shape. But then again, Clone probably smokes cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic tater tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So, at 50, you and Clone would probably look pretty different from one another. But it turns out, you don't just look different, you actually have become different. Now, if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that of the person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference, and if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely, potentially changing the message of that paragraph. The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern. And that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life. But these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary. And it can change over time, especially when your body is going through a lot of changes, like, say, during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're gonna have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're going to have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives, and it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very young science, though we've known about the epigenome since the 1970s. It's only in the last 20 years that we've even known what effect these epigenetic tags are having on our DNA. And even after they got all that business straight, scientists still thought that all of our epigenetic tags were stripped off of our genome before they were passed on to our children. So if you started smoking 10 packs a day when you were 10 years old, that would certainly be a horrible health decision for you, but you wouldn't necessarily be harming your unborn children in any measurable way. However, the thinking on that is changing pretty rapidly, because it's true that a lot, even most of the epigenetic information from a parent is stripped off of the embryo's genome in the first few days, and fresh ones are created specifically for this new person. However, some of these tags get stuck on the genome and are passed down from generation to generation. And it just so happens that the more they study this, the more it looks like bad epigenetic information is being passed from generation to generation. And this is a whole new way to think about how we pass information between generations. Your grandmother was making dietary decisions that affect you today. As we experience all of these new strange epidemics, diabetes, autoimmune disorders, cancers, that weren't appearing in previous generations, it's starting to look like these may be caused by epigenetic information passed down from our parents. I know, it's such an unbelievable buzzkill. There is no point in our lives when we can do anything without guilt anymore. The discovery that the environmental factors a parent experiences can be passed down from generation to generation was sparked in the 1980s. And this happened when some scientists were looking at the birth and death records of some people who lived in 19th century Sweden. This is a weird place to find a genetic revolution, but there it is. It wasn't just any place in Sweden. It was in Norrbotten, which is the northernmost county in Sweden, which is literally in the Arctic Circle. And despite the fact that Norrbotten was literally the worst place you could possibly choose to live in Sweden, there were some people living there in the 1800s. And they were completely cut off from the rest of the world. Let me clarify. These people were isolated. Like, if they didn't have a good crop year, people died. No, it wasn't ideal, but, you know, they didn't ask me my opinion, so I'm not giving it. So anyway, these people subsisted entirely on what they grew and the animals that they raised. And like I said, sometimes they starved. But sometimes they had huge, bountiful years of plenty. And when it happened, people totally went ape crackers. I mean, of course they did, because they were so freaking hungry, and then all of a sudden there was all this damn food everywhere. Anyway, there was a public health specialist who was looking at the effects of the people who grew up in the really bad starving periods of time versus the people who grew up in the eat all you can at the smorgasbord years. And you might already be guessing what they found out. People who went from relatively slim pickings to feed your face until you have to barf and then do it all over again in a single season, those people died an average of 6 years sooner than their starved out counterparts. And you know what sucks? So do their kids. And so do their kids' kids. Boo epigenetics. So, of course, now we all know, and we're all going to stop doing unhealthy things starting today. Unless the damage is already done. The damage is almost certainly already done. But hey, epigenetics brings good tidings along with the bad ones. For instance, we now know that certain types of cancer are caused by misplaced epigenetic tags, and scientists are now developing drugs that can silence the bad genes that were supposed to be turned off in the first place. Additionally, until recently, we thought that genes were the end-all and be-all of who you got to be. They were your blueprint, and you couldn't escape them. This outlook is not just kind of depressing, it also leads to a yucky sort of social prejudice. Because when you look at data without considering social and epigenetic factors, it might look like people with less money are less intelligent. So just like 15 years ago, there were scientists saying things in public, mind you, like, some people just have good genes for intelligence, and it just so happens that the poor people don't. Oh, it's so sad. Too bad, poor people. But you have bad genes. Well, it turns out that that is not even a little bit true. Not only are there a huge variety of social factors that affect how well people do on intelligence tests, but a genetic trait is also not just a product of genes, it's also a product of environment. Any one person's genome is determined by any number of decisions made by any number of their ancestors. And right now, you are making decisions that are going to affect people who are alive long after you're dead. No pressure or anything. And so I'm glad you've watched this, because when you and your clone are in your 60s and you sit down, have lunch, and he brings his son and his son has a tattoo on his face, he did not inherit that from you. This is Hank Green for The Sci Show. We hope you learned something.
Okay, so I think epigenetics is a very interesting concept to think about when we think about nutrition counseling, because I think it adds a whole nother level on to how we're interacting with people. So for instance, I mean, what responsibility does someone have if they're the genes they're expressing are due to the decisions made by the generations earlier? So I think it's really good to think about that this is just kind of one more piece of information of meet people where they are. We don't know what is happening inside with their genetic expression. So we really just need to meet people where they are and understand that there is going to be a lot of individual variability. Okay, so epigenetics does, yes, it applies to many different types of exposure. It could apply to pollution, for instance. But in this class, of course, and for us professionally, we're mostly interested in nutrition epigenetics or nutrigenomics. And this is the impact of nutrition on gene transcription. And we find in the literature that nutrigenomics could impact a number of the chronic diseases that we commonly treat in dietetics, such as cancer, aging, obesity, type 2 diabetes, inflammation, and neurocognitive disorders. And your readings for this week are on this topic. So here are your practice questions for this section. And now let's move on to our next section, which is the feed-fast cycle in protein metabolism and growth and development and injury and disease. So first, we're going to explain the 4 different stages of the feed-fast cycle. And this is really going to set the stage for a couple of lectures from now when we talk about the interrelationships between macronutrients in metabolism. But just today, we're going to just focus on protein. We're also going to describe the concept of the body's energy budget, identify changes in protein metabolism in growth and development, and in injury and disease. Okay, so the feed-fast cycle can be divided into 4 stages. And again, we will touch on the other macronutrients in these stages later in the course. So first we have the fed state, I just ate, the post-absorptive to early fasting state, which goes until about 12 hours past a meal, so think like overnight. The fasting state, okay, now we're overnight and maybe getting to be a little bit longer, and then the starvation state when you've gone without food for days. So first we have the fed state lasting about 3 hours after meal ingestion. And here is so again, we will come back to these figures, so don't worry too much right now about the other macronutrients. Let's focus specifically on protein and amino acids here. So here is the disposition of dietary amino acids in the fed state. So here we have amino acids from the gut. Amino acids go to the liver. They can be deaminated to become pyruvate and then be built in triglycerides and go into circulation and also be formed into adipose tissue. Amino acids can go into portal circulation and go to other tissues such as the CNS or to our muscle. And that's it. So that's what predominates in the fed state. Next, let's move to the post-absorptive state. So it goes from about 3 hours till about 12 to 16 hours following a meal. So think like over a long stretch between meals to overnight. So at this point, tissues can no longer derive energy from ingested glucose and must depend on other sources for fuel and from glycogenolysis and gluconeogenesis. So it is in this stage that the alanine glucose cycle begins to become important. Let's look at this video. I can't remember exactly what this one is.
The day after chemo, you could be home with your support.
For more information on tutoring, personalized video solutions, or how to support MOOF University, and the production of more videos, check out moofuniversity.com. Thank you and enjoy. Okay, so previously we saw that ammonium ion can be transported safely from tissues to the liver by way of glutamine. Glutamine being the main sort of amino acid that carries nitrogen through the blood and around the body. Now, that doesn't have to happen exclusively with glutamine. It can also occur with another amino acid, alanine. And that alanine is specifically coming from the skeletal muscles. Okay, so what's going to be going down is that in the muscle we're going to be breaking down proteins, and we'll basically take amino groups as we're basically taking amino groups from the muscle out through the blood to the liver. And we're going to have a little cycle here, and we're going to see what that's going to look like. Okay, so we'll start off in a skeletal muscle cell. We'll make our way through the blood and eventually to the liver. So let's start here in the skeletal muscle cell with glucose. So in the skeletal muscle cell, we're going to be talking about taking glucose and metabolizing it to pyruvate via glycolysis. Once we get that pyruvate will undergo a transamination reaction catalyzed by alanine aminotransferase. And the reason it's named alanine aminotransferase is because it's going to turn that pyruvate into alanine, because alanine is the alpha amino acid counterpart to pyruvate being an alpha keto acid. So where's that amino group coming from? Well, it's coming from the transamination reaction, specifically from glutamate. Where is that glutamate coming from? Well, we're breaking down proteins in the skeletal muscle into amino acids, and of course those amino acids have nitrogens on them. Those will end up on glutamate, and that glutamate can supply that nitrogen for this aminotransferase reaction. And so what we're going to do is we're basically taking this nitrogen here that's a breakdown product from those proteins and tacking it onto pyruvate to make alanine. Now that alanine that's produced can be sent in the blood through to the liver. And once it's in the liver, it'll basically undergo that same aminotransferase reaction, that same transamination reaction to get pyruvate back. And what happens to that amino group? Well, it's undergoing this whole thing is happening as a transamination reaction opposite what occurred in the skeletal muscle cell. That alanine will be turned into pyruvate, and this amino group will end up being attached to alpha ketoglutarate to produce glutamate, except all of that glutamate is in the liver. Oops. So now what can happen with that glutamate is you can undergo the glutamate dehydrogenase step to free up that ammonium ion, which of course can be excreted safely as urea after going through the urea cycle, or that nitrogen can be used to supply transamination reactions to produce aspartate, which can also go through to the urea cycle. The point is, though, that we're basically getting the nitrogens that are sort of the products of breaking down proteins in the muscles out into the liver, right? So we're taking, let's do that in a different color. We're taking these guys out through the blood and to the liver so that they can eventually be excreted safely in urea. Now, the whole situation that we're talking about is the glucose-alanine cycle, but I haven't actually talked about a cycle quite yet. I haven't finished it. I've just talked about it one way, going from the skeletal muscle to the liver. Once this pyruvate is produced in the liver, it can undergo gluconeogenesis to produce glucose, which can be sent back to the skeletal muscle via the blood, right? So that that glucose can be broken down for energy in the skeletal muscle cell. So the point here basically is that the ammonium ion from extrahepatic tissues is not only transported by glutamine. That's one point here. It can also in muscle tissue, alanine can do the job, especially because this contributes to getting glucose back to the muscle tissue, so that skeletal muscle tissue can go through and metabolize that for energy. Now this sort of schematic might look kind of familiar, kind of the muscle and liver kind of with a cycle. If you recall, there is another cycle, the Cori cycle, that does a sort of similar thing. This works well with the Cori cycle basically to get glucose back to metabolically active tissue. And we're kind of killing two birds with one stone. In addition to doing that, you're getting the nitrogen out from the skeletal muscle to the liver to be detoxified. Okay, so that's basically the glucose-alanine cycle. I hope that video was helpful.
Okay.
Thank you for watching.
So with the glucose-alanine cycle, we're able to break down protein from the muscle for energy, get the nitrogen out of the muscle to the liver where it can be excreted in the form of urea, and then we send the glucose back to the muscle so that the muscle can use it, can again break it down to pyruvate. And just think about that this process, glycolysis of converting glucose to pyruvate, is a much faster reaction to produce ATP than going through all of oxidative phosphorylation, TCA cycle, electron transport chain. So in this way, we're getting a quick source of energy, and we're also able to get rid of the nitrogen in a safe way through the liver. So again, this process begins to be important in the post-absorptive or early fasting stage. We're just starting to kind of start breaking down our muscle a little bit to use it for energy. So branched chain amino acids are utilized as fuel at this stage, and other amino acids are released in lesser quantities. So again, so this is in the post-absorptive phase, and again, let's just concentrate on the amino acids. And it's these dark lines that are the processes that predominate in this stage. So we can see here that we have our alanine from our muscle breakdown, and the alanine is going to go to the liver. The glucose from the liver is going to go to the muscle, and we have our glucose-alanine cycle. Next, we'll move to the next stage, which is the fasting stage, and this lasts up to 2 days without any additional food intake. And now we're really kicking up the breaking down our muscle protein for gluconeogenesis. So some of our substrate for gluconeogenesis is going to come from the glycerol and lipolysis. Some is going to come from lactate and anaerobic glucose metabolism, but a lot of it's also going to come from muscle breakdown. So of all of the amino acids, only leucine and lysine cannot contribute to gluconeogenesis because we know that they are the only amino acids that are exclusively ketogenic. So they can be converted to ketones for other energy uses, but they cannot contribute to glucose production. Now, during prolonged fasting, we have low glucose levels, and this leads to a glucagon hormone and corticosteroid hormone predominating, shifting our body to gluconeogenesis to produce the glucose that we need for our body processes, such as brain function. Okay, now, if an individual has not had food for several weeks in duration, then they move on to the starvation state. So, in the beginning, remember, in the fed state, we're just using the amino acids and the glucose that we got from our meal. Then we are starting to kind of break down our muscle a little bit for gluconeogenesis. Now, once we progress to the starving state, our body does a really smart thing. It shifts again and starts conserving muscle protein so that life can continue to function. Think about it. We need muscle functioning to breathe. We also need antibodies to fight infection. We need our enzymes. We need hemoglobin for oxygen transport. So once our body starts to realize this is serious, we do not have any food coming in, then it starts to really conserve this muscle protein. And we have more of a metabolic shift towards sparing body protein and emphasizing more lipolysis so that we can use the energy from lipolysis. So during the starvation stage, fat becomes our major energy supplier. Okay. And as we talked about in the lipids section, we know that fatty acid catabolism in times of food deprivation, and especially glucose deprivation, leads to production of ketone bodies. And then these can be exported to our muscle tissues, including our heart, and to our brain tissues for oxidation instead of glucose, thereby retaining as much glucose as possible. So as long as ketone bodies are remained at high levels, then the need for glucose and gluconeogenesis is reduced, and therefore our body protein is spared. And so our survival time once we enter the starvation stage depends on what our fat stores were before starvation. Sorry, this is kind of not super clear, but this is the flow of substrates during the starvation phase. And again, the solid lines are the ones that predominate. So again, there is a shift during starvation to fatty acid and glycerol export from adipose tissue and increased fatty acid and ketones as fuel by the CNS and the muscle. And we have less breakdown of our body proteins at this stage. Okay, so again, we're kind of starting to wind up everything and pull everything together. So you'll see some of this more in a couple of lectures, but I do want to just touch on it right now. So here is a summary of the metabolism of insulin and glucagon. And right now, let's focus on the amino acids. So when we have insulin, it's going to, the production stimulated by, so insulin is produced and stimulated by rising blood glucose and amino acids from the diet. So amino acids can cause insulin release. And this causes decreased gluconeogenesis in the liver, increased amino acid uptake in the muscle and most other tissues, and increased protein synthesis in the muscle and most other tissues, which makes sense. We've got plenty of amino acids, let's build them into something. When glucagon predominates, such as in the starvation phase, first we know that its production is stimulated by low blood glucose, and we know that it's going to cause increased gluconeogenesis in the liver and increased amino acid uptake in the liver for gluconeogenesis. Now let's talk about the body's energy budget. So let's rewind. Let's go back to the fed state, feasting. So when a person eats in excess of energy demands, they have surplus protein. Now we know extra protein isn't stored in the body as protein. We can't grow bigger muscles by eating a ton of protein powder, right? Well, at least not by only eating a ton of protein powder. So first, when we eat as many amino acids as we need, the body's going to replace our normal losses, that such as the sloughing of cells in our digestive tract. And then it's going to increase protein oxidation slightly. So remember, insulin's predominating. We have tons of amino acids. What are we going to do? Build up, protein synthesis, break down, protein oxidation. So any additional protein is going to be deaminated and remain as a carbon skeleton, and then it can be used to make fatty acids. So a person can gain adipose tissue by eating too much protein. Now we move on to between meals and overnight fast, post-absorptive or early fasting stage. So between meals, the brain and liver can still rely mainly on glucose for energy. Our fat stored in adipose tissue can provide free fatty acids to the liver and be broken down for the energy. And because our bodies are relying on carbohydrates and fats, we really have this sparing effect on our body protein. They don't need to be used for energy. However, when we extend this phase to an overnight fast, then we are starting to rely on this glucose-alanine cycle. I'm breaking down a little bit of our muscle for energy, but it's such a small amount that there's really no negative effect on our muscle mass or function by just fasting overnight. Then we move to fasting. And here, the muscle proteins are providing amino acids to be broken down by energy. And this could occur during severe weight loss, severe dieting, early eating disorders, etc. And one thing that we need to think about when we're relying on breaking down our muscle proteins for energy is that it's a little bit more complicated than when we break down just carbohydrates and fats. So when we're breaking down carbohydrates and fats, we're dealing mostly with carbons, hydrogens, and oxygens, and there's really no complications or difficulties for the bodies to break these down. However, amino acids have a nitrogen component, and we know that if we have too much nitrogen or ammonia in our blood, that it can be toxic. So our body, when we break down protein to use for energy, we know from last week that we've got to go the extra step to transaminate or deaminate these amino acids so that we can use them for energy. So this is just one more step that the body needs to take. Now, we know that protein synthesis is going to be altered depending on our physiologic state. So first, let's talk about normal growth. We know that growth is an anabolic process that logically includes a net increase in body protein. So often this is with significant remodeling, so that protein degradation is also elevated. So think of it as, yes, I'm building protein 2 steps forward, but I'm also degrading protein 1 step back. But the net is an increase in body protein. So the younger we are, the faster our rates of protein synthesis. In infants, it's 2 times the rate as we see in adults. However, this rate of protein synthesis declines through time, throughout the life cycle. Now, we do have kids that experience malnutrition, and we know that from that, they can experience wasting or stunting. But if we are able to refeed children, then they actually have a faster rate of growth and rate of protein synthesis, and that's why we call this catch-up growth. Because if some children, if they haven't been deprived of food for way too long, then they can bounce back at least to a certain extent. And this catch-up growth is accomplished by an increased levels of RNA in the tissue, which then, as we saw from earlier in the lecture, equates to more protein synthesis. Now, we also have growth during puberty, during pregnancy and lactation, and during these stages, we have alteration in our hormone concentrations, and that can impact our protein biosynthesis. So, for example, we may express androgen, which is anabolic, and that can provide increase in protein mass. Okay. Now, when individuals experience malnutrition, as we know, there's an increase in glucagon levels, and our growth hormones are elevated. So think about it, that our body is saying, in times when a child is trying to grow, for instance, okay, we need to grow, we need to build proteins, we need muscle, and obviously you're not doing that, so I'm going to up my growth hormone. Now, at this same time, thyroid hormone levels are reduced with a similar conservation of energy and amino acids as seen in starvation, because this is starvation. Maybe these individuals are getting some food every day, but if you look at the big picture, they're getting such an inadequate amount that it is considered a form of starvation. Now, during this stage, oxidation of amino acids is reduced, which makes sense. Just like in starvation, we're trying to conserve our body protein, and we decrease protein turnover. In children, this is accomplished through growth cessation, such as in stunting, where we have a reduction in height for age, in wasting, where we have a reduction in weight for height. In malnutrition, we also see that albumin concentration is often reduced, since our body is trying to conserve proteins, our amino acids, and only use them for the most essential functions. Now, I think many of you are exposed to these physical exams for protein or for malnutrition, so I'm just going to go over it very closely. But an increasing trend in dietetics is the physical exam for malnutrition. And there's specific points where the dietitian is supposed to assess both visually and perhaps through touch physically in order to assess subcutaneous fat loss and muscle loss. So here, I'll let you read through these yourself, but for instance, we would have for subcutaneous loss, the orbital region surrounding the eye, and we have different classifications of malnutrition according to what they look like. And all of these different pieces kind of go together to make a whole picture so that we can assess malnutrition. And here's just some examples. So you can see that in the chest, thorax region, that you can see bones here. This would be indicative of wasting. We can see ribs in the back here, shoulders here, clavicles protruding. So kind of look through the chart and look at the pictures and just kind of try to get an idea of how we would recognize malnutrition in our clients. And hopefully you'll be able to practice this in the real world too. Okay, now let's shift from growth and development to injury and disease. Now, loss of body protein accompanies many diseases, such as trauma, cancer, and infections. And these are in the biological processes that our body undergoes in regards to protein synthesis and degradation do contrast malnutrition and starvation in that The adaptation that we see in starvation and in malnutrition, that adaptation that we see to conserve body protein, that doesn't happen here in injury and disease. So really, the body does not have time to adapt to preserving the muscle mass. So a person can lose a lot of muscle mass very quickly. And in this table, we have comparison and contrasting. So whereas in malnutrition, starvation, we would preserve lean body mass, we don't have this adaptation, injury, and disease. Whereas we might have lower metabolic rate in malnutrition and starvation, we may be hypermetabolic in injury and disease. And in starvation, we saw that we shift so that we have a higher dependency on ketones so that it's protein sparing. And again, we do not have this adaptation in injury and disease. And here is a figure demonstrating the pathophysiology of protein energy malnutrition. So we have inadequate intake. Normally, we would have adaptable mechanisms and successful adaptation. However, if we have from injury and disease, metabolic stress, or our starvation is way too severe, we can have failed adaptation with the consequences outlined here, including immune deficiency. And here are your practice questions too. And that is all that I have for you today. So we will talk next week about in more detail about the role of protein in health and disease. And I hope you all have a great week.